My name is Jihyo Lim, and today I'll be presenting about photosynthesis regarding the topic of biology. So photosynthesis is defined as the conversion of light energy into chemical energy. In simple terms, plants take sunlight and turn it into food in the forms of sugars. This process takes place in the leaves, specifically in structures called chloroplasts. A chloroplast is an organelle, meaning a specialized structure inside a cell that performs a certain function. On the surface of leaves, we also find small pores known as stomata. These pores allow carbon dioxide, or CO2, to enter and oxygen to leave. In summary, photosynthesis is the process by which plants capture sunlight, transform it into energy, and release oxygen, a process essential for life on Earth. Photosynthesis is a type of redox reaction. The word redox stands for reduction and oxidation, which means that electrons are being transferred between molecules. Oxidation means losing electrons. Here, 6 water molecules are oxidized to 6 oxygen molecules. Reduction means gaining electrons. Here, 6 carbon dioxides are reduced to glucose or sugar. The entire process of photosynthesis can be divided into 2 main stages. The light reactions, where sunlight is captured and turned into energy molecules, and the Calvin cycle, where that stored energy is used to make sugar from carbon dioxide. The first stage is the light reactions, which occur in the thylakoid membrane inside the chloroplast. Here, solar energy is converted into 2 types of chemical energy. ATP, short for adenosine triphosphate, which is the main energy carrier in cells. NADPH, which is another energy molecule that carries electrons. During this process, sunlight excites electrons. Think of electrons as tiny particles inside atoms. When they're excited by light, they gain energy, and that energy is used to create ATP and NADPH. Therefore, the light reactions essentially transform sunlight into usable chemical batteries that fuel the next stage. Let's now take a closer look at how ATP is generated. This occurs through a process called chemiosmosis. In this process, sunlight provides energy that pumps protons or hydrogen ions across the thylakoid membrane. These protons then flow back through an enzyme known as ATP synthase. As they pass through, the enzyme uses their movement to create ATP from ADP. In this way, the energy from sunlight is captured and stored in the bonds of ATP molecules. The second stage of photosynthesis is called the Calvin cycle. Unlike the light reactions, this stage does not require light directly. Instead, it uses the ATP and NADPH generated earlier to convert carbon dioxide into sugar. It takes 3 full cycles of reactions to produce 1 sugar molecule. The Calvin cycle has 3 main phases: carbon fixation, reduction, and regeneration of RUBP. So first, carbon fixation. In this step, the cycle begins when 3 molecules of carbon dioxide enter. Each CO2 molecule attaches to a 5-carbon compound called RUBP. This reaction is catalyzed by an enzyme called rubisco. The result is an unstable 6-carbon compound that immediately splits into 2 3-carbon molecules known as 3-phosphoglycerate or 3-PGA. In this stage, carbon from the atmosphere has been fixed or captured into a solid organic molecule. Second is reduction. Each 3-PGA molecule is transformed into a higher energy molecule called G3P. This step requires energy. Specifically, ATP donates energy and NADPH donates electrons. Through this reduction process, the carbon molecules gain energy and are converted into sugar precursors. Out of the 6 G3P molecules produced, only 1 will exit the cycle to become part of a sugar, while the remaining 5 are kept in the cycle. And third are regeneration of RUBP. The final step uses ATP to rearrange the remaining 5 G3P molecules back into 3 molecules of RUBP. This regeneration is crucial because it allows the cycle to continue capturing new CO2 molecules. Now let's take a look at practice problems using the knowledge we just learned. First question is, how are the Lye reactions and the Calvin cycle connected? The correct answer is B. The Lye reactions provide ATP and NADPH to Calvin cycle, and the Calvin cycle returns ADP, phosphate, and NADP+ to the Lye reactions. The Lye reactions and the Calvin cycle are tightly connected and dependent on each other. The Lye reactions produce ATP and NADPH, which are the energy carriers and reducing agents needed for the Calvin cycle. The Calvin cycle then uses up those molecules to build sugars and in the process converts ATP back into ADP plus phosphate and NADPH back into NADP plus. These recycled molecules return to their Lye reactions where they are re-energized. Next question is, if you wanted to design a very specific chemical to target the Lye reactions of photosynthesis, where should the chemical be designed to go most specifically? The correct answer is B. Thylakoids. The Lye reactions of photosynthesis occur in the thylakoid membranes inside the chloroplast. This is where photosystems, protein complexes that capture light, the electron transfer chain, and ATP synthase are all located. These structures work together to convert sunlight into ATP and NADPH. Therefore, if you wanted to design a chemical that specifically interferes with the Lye reactions, you would need to target the thylakoid membrane, not the stomata or mesophyll. Finally, let's discuss the importance of photosynthesis. First, foundation of the food chain. Plants are the producers that provide energy for all other living organisms. Next, oxygen production. Photosynthesis releases the oxygen we need for respiration. Third is climate regulation, which is plants absorb carbon dioxide, helping to balance our atmosphere and slow global warming. Lastly, energy for ecosystems. All the energy in food webs originates from photosynthesis. In conclusion, photosynthesis is not just a plant process. It is the foundation of life on Earth, providing food, energy, oxygen, and climate stability. Without it, neither humans nor most organisms could survive. Thank you.